Good morning, students. Welcome back to biology class, chapter 13, photosynthesis in higher plants. All animals, including human beings, depend on plants for their food. Green plants make or synthesize the food they need, and all other organisms depend on them for their needs. Green plants carry out photosynthesis. They are using the light energy to synthesize the organic compounds. All living forms on earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basis of life on earth. Photosynthesis is an enzyme regulated anabolic process of manufacture of organic compounds inside the chlorophyll containing cells from carbon dioxide and water with the help of sunlight as a source of energy. Photosynthesis is a chemical process uses light energy to synthesize organic compound like sugar. Photosynthesis is important due to two reasons. First one, it is the primary source of all food on earth. It is responsible for the release of oxygen into the atmosphere by green plants. Now we'll see that one, what are the experiments done for what are the factors affecting for the photosynthesis. In the early experiments, Joseph Priestley in 1770 performed a series of experiments with a bell jar, candle and mouse. Priestley observed that a candle burning in a closed bell jar soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damaged the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive in the candle, continued to burn. Priestley hypothesized as follows: plants restore to the air whatever breathing animals and burning candles remove. Check in the diagram. Jan Ingenhousz 1732-99 showed that sunlight is essential to the plant process that somehow purifies the air fouled by burning candles or breathing animals. He performed experiment with an aquatic plant, showed that in bright sunlight small bubbles were formed around the green parts, while in the dark they did not. Later, he identified these bubbles to be of oxygen. Hence, he showed that it is only the green part of the plant that could release oxygen. In 1854, Julius von Sachs proved that plants produce glucose while growing and stores them as starch. He explained that plants have green substance which is present within a cell in specialized bodies. He found that the green parts in plants is where glucose is made and that the glucose is usually stored as starch. T.W. Engelmann split light using prism vibgeo, green algae cladophora placed in a suspension of aerobic bacteria. Bacteria were used to detect the sites of oxygen evolutions. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. He explained the absorption spectra of chlorophyll A and B. Key features of photosynthesis was first explained in 19th century that plants could use light energy to make carbohydrates from carbon dioxide and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organisms was then understood. It's represented as a carbohydrate. Carbon dioxide plus water in the presence of light, they are getting the carbohydrate. Then Cornelius van Niel, he did the experiment with the purple and green bacteria and demonstrated photosynthesis is a light dependent process. He explained hydrogen from water reduces carbon dioxide to carbohydrates. He concluded that oxygen comes from water and not from carbon dioxide. Finally, the correct equation for photosynthesis was discovered. Next we'll see what is the site for photosynthesis, where the photosynthesis will take place. Photosynthesis will take place only in green parts of the plant, mostly in the leaves. Within a leaf, photosynthesis occurs in the mesophyll cells which contain the chloroplast. Chloroplasts are the actual sites for the photosynthesis. In the chloroplast, thylakoids contain most of the pigments required for capturing solar energy to initiate photosynthesis. The membrane system, that is the grana, is responsible for trapping the light energy and for the synthesis of ATP and NADPH. Biosynthetic phase, that is the dark reaction, is carried in stroma where carbon dioxide is reduced to carbohydrate. Now we'll see the division of labor within chloroplast. The membrane system grana is responsible for trapping light energy and synthesis of ATP and NADPH. These light-driven reactions are called light reaction. In stroma, series of enzymatic reactions lead to synthesis of glucose from carbon dioxide. These reactions are not directly light-driven but are dependent on products of light reactions like ATP and NADPH. These reactions are called dark reactions. However, it does not mean that dark reactions occur in darkness or they are not light dependent. Pigments involved in photosynthesis. Chlorophyll A, bright or blue-green. Major pigment act as a reaction center. Chlorophyll B, yellow-green. Xanthophyll, yellow. Carotenoid, yellow to yellow-orange. In the blue and red regions of spectrum shows higher rate of photosynthesis. Photosynthetic pigment can be found in thylakoid and intergranal lamella. To absorb the visible light, photosynthesis pigments are the substances that absorb visible light. Narrow band between 380 to 750 nanometer is the function of this pigment. Each wavelength is different colored light. The efficiency of photosynthesis in different wavelengths can be explained by considering the absorption properties of the photosynthetic pigment. The absorption spectrum of a pigment, chlorophyll A and chlorophyll B, show that absorption is higher in blue 445 to 500 nanometer and red 620 nanometer to 740 nanometer light. The least is green 500 to 575 nanometer in the light. Wavelength light that optimally absorbed and used by green plants as a source of energy are the blue and red regions. A chromatographic separation of leaf pigment shows that color in leaves consists of 4 pigments: chlorophyll A, blue-green, chlorophyll B, yellow, xanthophylls yellow, and carotenoids yellow-orange. These pigments absorb light at specific wavelength. Maximum absorption light occurs in blue and red regions of spectrum by chlorophyll A. Rate of photosynthesis also higher in blue and red regions. Conclusion: chlorophyll A is the chief pigment associated with photosynthesis. Chlorophyll A is the most abundant plant pigment in the chloroplast. And though chlorophyll A is the major pigment responsible for trapping light, other thylakoid pigments like chlorophyll B, xanthophylls, and carotenoids, which are called accessory pigments, absorb light and transfer the energy to chlorophyll A. Accessory pigments enable a wider range of wavelength of light to be utilized for photosynthesis, protect chlorophyll A from photooxidation. The portion of spectrum between 400 nanometer to 700 nanometer is called photosynthetically active radiation. What are the differences between photosynthetic pigments? Like chlorophyll A act as primary pigment, participate directly in light dependent reaction, convert light energy to chemical energy. Chlorophyll B act as accessory pigment, captured light and transfer it to chlorophyll A, then initiates the light dependent reactions. Thank you.